Okay, this is a presentation on Neo-Mendelian genetics to accompany the work that you'll be doing in the virtual fly lab. Neo-Mendelian genetics was developed by Thomas Hunt Morgan working with Drosophila melanogaster, the fruit fly you see here. So we should first talk about Mendel's good luck. You know, Mendelian genetics was a monumental achievement where he developed these postulates that explain the inheritance of traits so cleanly, but not all traits are inherited in the ways that he modeled. Perhaps he ignored some other traits that didn't comply with his simple rules, or perhaps he was just lucky. The traits that he was looking at were traits where each character was determined by only a single gene. There weren't multi-gene traits, and there were only two alleles for each gene, not multiple alleles. And one allele was completely dominant to the other. There was an example of incomplete dominance, like in pink carnations. And the alleles were on the autosomes, not on the dimorphic sex chromosomes. And whenever you look at these traits, the classic ones that are used in the textbook to introduce Mendelian traits like purple and white flowers and round and wrinkled seeds, you get these really clean ratios in the offspring of your crosses. Like in the monohybrid cross, you get a 1:0 ratio, or 100% purple, let's say purple flowers in the F1 generation, 3:1 ratio in the F2s. When you do your test crosses, a 1:1 ratio. Likewise with dihybrid crosses, 100%, like those round and yellow seeds. And 9:3 to 3:1 ratios in the dihybrid cross, and your test cross is a 1:1 to 1:1 ratio, so very clean. But Thomas Hunt Morgan was a geneticist, or was becoming a geneticist, and was using Drosophila and not pea plants as the model organism. Was able to duplicate a lot of Mendel's research and find out that there are a lot of traits that are inherited according to Mendel's simple rules. However, when he found some mutants that weren't inherited according to Mendel's simple rules and gave different ratios, he explained these exceptions, and we call this extension of Mendelian genetics Neo-Mendelian genetics. So let's take a look at some of these Drosophila mutants that arose in the lab. So we've got all kinds of different body colors, all kinds of different wing shapes. And one thing I like to ask my students is, can you find the wild type fly? Well, this fly here is the wild type fly. As you can see, it shares a lot of the characteristics with the other fly, each of which differ from this fly by one trait. Here we've got an ebony-bodied fly instead of a tan-bodied fly, a yellow-bodied fly instead of a tan-bodied. We've got one with short wings, curly wings, different eye colors, orange eyes, and white eyes. So I guess we need to talk a little bit about the fruit fly as an organism. It's familiar to lots of us. We find it around rotting fruits in our own houses. The fruit fly is sexually dimorphic. The females are a little bit larger than the males, and the males, you can identify them by this kind of dark place at the rear end of their abdomen. The life cycle is pretty short, and that's the reason that they're used as a model species in genetics. It only takes about 14 days to get through the life cycle. After an egg is fertilized, it sort of moves through different larval stages, then pupates and metamorphosizes into a fly. And like I said, that only takes 14 days. So you can do a lot of genetics in a hurry with fruit flies. Also, each individual fly produces many offspring. But we'll actually be accelerating our genetics experiments even faster by doing a virtual fruit fly lab on the computer. So I need to talk about something. I need to talk about the notation that is used to describe phenotypes and genotypes in fruit flies. It's a lot different than the Mendelian notation that you're familiar with. Big P for the dominant, say, allele for purple flowers, and little p for the white flowers. And this is just the way that Thomas Morgan did it in his lab. Okay, so first of all, this is a wild type fly on the right, and this is a mutant fly, an ebony-bodied mutant on the left. Now, that ebony-bodied mutant fly we could call mutant, we could call ebony, or capital E. You'll notice that the phenotypes are in capital letters. It doesn't mean dominant, it just means phenotype. And the phenotype for the wild type fly, or the normal looking fly, is wild or plus. Now the genotypes are small letters, and here again the small letters doesn't mean recessive, though it could be. It doesn't mean recessive, it just means genotype. So here we have little e for ebony, and this is a homozygous individual. There's two alleles for this genotype. Now for the genotype of the wild type fly, we've got E+, or EE+. Now plus means the wild type version of the allele that, if it were mutated, would be the alternate trait. Sounds a little confusing and backwards, but you'll get the hang of it. So the reason that we have two genotypes here is, I'll let you all think about that one for a second. It's because the fly could either be homozygous dominant or heterozygous. And so in this case, the wild type is dominant over the ebony-bodied trait, and so E+, which represents the wild type allele in the homozygous dominant individual, there's two of them. And there's one recessive allele, which is the mutant allele specified by E, and then E+ is the wild type version of that, gene. Okay, so the notation's a little bit different, but I'm hoping that you'll get the hang of it soon. Okay, so the virtual fly lab is a computer simulation where you'll go on and you'll order flies and cross them. And in crossing them, I want you to you will only see the phenotypes. It's kind of cool that way, just like in the real world, you only see the phenotypes of the fly. You can't do any sort of extraction of DNA or gel electrophoresis. But in order to understand what's going on at the level of the genotype, you have to infer it, much like early geneticists did. So the first three experiments in the virtual fly lab focus on classical Mendelian genetics. So we'll reproduce Mendel's monohybrid cross by crossing a wild type female fly which has red eyes with a mutant sepia-eyed male fly. And then you can take that to the F2 generation and see what ratios you get, 3 to 1. Let's see, and sort of confirm that Mendel's simple rules also work in these animals. In experiment number 2, you'll take an individual that's in the F2 generation that has a known phenotype but an unknown genotype. It could either be, I guess we can go back to this picture here, it could either be E+, well, it wouldn't be ebony here, it would be SE+, SE+, or SE, SE+, and determine through a test cross what the genotype is. Then in the third experiment, you'll be doing a dihybrid cross, so crossing individuals that are ebony-bodied with an individual that has vestigial wings. You see these little wings are kind of shrunken and vestigial, and you'll get in the F1 generation there'll be like a double heterozygote, and then you'll cross that one to see if you get the ratio that you're looking for in the classic dihybrid cross, 9 to 3 to 3 to 1. Okay, so in experiments 4 to 7 is where we actually get to this Neo-Mendelian genetics that I was talking about before. These are, genes and alleles and traits that Mendel wouldn't have been able to explain with his simple ratios, and Morgan had to sort of get a better understanding of what was going on at the genetic level and create new rules to describe these odd mutations. The first will be a sex-linked trait, where you'll be crossing a white-eyed fly with a red-eyed fly, then the wild-type red-eyed fly. In the experiment number 5, you'll be finding a mutant, so you'll be using trial and error to try to find a mutation that is inherited like sepia eye, like the simple Mendelian trait. In the case of the lethal allele, you'll be looking at this Aristopedia antenna, which has a very interesting trait in that it's lethal when homozygous. And for number 7, you'll be looking at a case of epistasis, which is where one gene blocks another. And in this case, we'll be trying to determine, does vestigial wings block incomplete veins, or does incomplete veins block vestigial wings? So let's take a closer look at the sex-linked traits. The white-eyed mutant was one of the first to be identified by Thomas Hunt Morgan, and what Morgan found was that when he crossed a white-eyed male with a red-eyed female, you got all red-eyed offspring. So that's not so surprising. I mean, this is kind of like, purple flowered plants and maybe white plants, and you get all purple flowered in the next generation. It's not so surprising to get the dominant trait. But if you flip the script and you cross a white-eyed female with a red-eyed male, you actually get all red-eyed daughters and all white-eyed sons. So it appears that the trait is linked to sex in somehow. And he explained it by hypothesizing that there were dimorphic sex chromosomes, that there were different sex chromosomes that the different genders had, that females have 2 X chromosomes and males have an X and a Y chromosome, and the gene for this eye color is on the X chromosome. So if males inherit a single copy of, either allele, they are hemizygous for that particular trait. So when you look at the Punnett square here for this particular cross, we have a white-eyed mother that donates a copy of the white-eyed allele in the egg cells, and then the father in sperm cells, half of them have an X chromosome with the red-eyed allele, and half of them are the Y chromosome, which doesn't have either the red or the white allele on it. And so all the daughters get one of each, white and red, and therefore they're heterozygous. And the sons receive a Y chromosome from dad, and then the chromosome that they receive from mom is the white-eyed. And so since they're hemizygotes, they express the white-eyed trait. And so the gender of the offspring really is linked to the trait that they exhibit. So this was certainly something that Mendel didn't encounter, and Morgan explained it. All right. So the way you need to write these genotypes for these phenotypes is, for a white-eyed female, you would write XWXW for the mutant version of both of those alleles. For a wild-type heterozygous female, you would write, a XW plus for the wild type, and this is heterozygote, so XW. For the male, it would be XWY, and for the white-eyed male and for the wild-type male, XW plus Y. All right. So lethal alleles. So the example that's often used for lethal alleles is this case of the Mexican hairless dog. Here these two dogs are the same breed, just that one of them doesn't express any hair and the other does. And in our case, we'll be looking at Aristopedia as a lethal allele, and we'll be looking for this ratio of 2 to 1. Let me explain how that works. So a lethal allele is an allele that if you have one copy of it, it results in a weird trait like not having any hair. But if you have a double dose of it, it could actually kill you, or it kills an embryo so it doesn't even really develop. So in this case, the lethal allele is actually dominant. So it's, in this case, using Mendelian notation here, it's big H. The alternative allele is the wild type, which is little h. And let's look at the genotypes. So if you have no hairless alleles, then you're a hairy dog. So you're little h, because it's the lethal allele is actually dominant. If you're a heterozygote, you're hairless. And if you have a double dose, then you die. And so as a result, you're never exhibited in the offspring. So when you cross two hairless dogs, it's like you have a cross between two heterozygotes, and you would get a 1 to 2 to 1 ratio among the genotypes and a 3 to 1 among the phenotypes, but 1/4 die off. So then you get this 2 to 1 ratio. And I don't really need to cover this, but if you were to cross a Mexican hairless and a hairless, it'd be like a test cross, so you get a 1-to-1 ratio. Okay, so just to back up, you'll be doing this with a trait which is thought to be lethal, the Aristopedia antenna. Okay, now epistasis is this interesting trait or condition where one gene blocks another. Let's consider two genes, B and C, in mice. And the gene B determines whether or not the mouse is going to be black or brown. If you get a double dose of the black allele, the dominant allele, you're going to be black. If you get only one, you're going to be black. Only when you have 2 copies of the recessive little b allele are you brown. Okay, so that makes sense. But then there's this other gene, this gene C, that affects the pathways that actually produce pigment in the first place. So if you have a double dose of the recessive allele, little c, you actually don't produce any pigment at all. You're white. And so you don't really know if the mouse would have been brown or black. That's when I say that one gene masks another, the gene C masks gene B, because you don't know if the individual was homozygous or would have been brown or black. All right. Okay, so normally when you cross 2 double heterozygotes, you get a 9-to-3 to 3-to-1 ratio, but because this C masks B, all 4 of these individuals in your outcomes in your Punnett square are white, so you get a 9-to-3 to 4 ratio. In the fruit fly example, you'll be looking at vestigial wings and incomplete veins, and you'll see if you get a 9-to-3 to 4 ratio in your crosses. And then you have to kind of interpret those by looking at the numbers or just by looking at the phenotypes themselves and you answering the question, is it vestigial wings that masks incomplete veins or incomplete veins that masks vestigial wings? So in the next video, I'll be explaining how the virtual fly lab works.